give you a little amuse-bouche, if you will, about how to work with EHR data. Going to give you a couple of practical hands-on skills. So we are going to, if I can get out PowerPoint to work with me, we're going to give you a brief introduction to electronic health records at all. For those of you that are clinical, you work in this area every day, but I'm going to be talking about EHRs from the perspective of how to use them for data, which it can be a little bit different than how you work with them as an end user. Then we're going to dive into what's the landscape of EHR-based research, thinking about the different kinds of methods that are available for you to use. We're going to go through one of those methods, what's called a phenome-wide association study, and talk about what it is, how to do it, and when it might be appropriate to use for your research question. And finally, we'll do a deep dive into computational phenotyping, which is my methodologic area of expertise, but really is at the heart of how to make EHR data usable for research. And finally, we'll end on a list of CE resources, give you some practical tips on how you can learn more so that you can continue this journey into EHR-based research if you are interested. So with that, let's get going. So what is an EHR? If you're a clinician, you probably are cursing the EHR. If you're a patient, you may have looked over your doc's shoulder and seen a copy of Epic. And so this is what an EHR looks like. There's usually a patient. There's different views about the patient. There are things like, clinical notes. So as you go to an appointment, your doctor or provider needs to fill out, why did they see you? What's going on? What's your past medical history? What's their assessment of your clinical condition and their plan for how to treat you moving forward? There's all kinds of structured data, like what kind of medications have you been prescribed? There are some lab testing data that's available. Obviously, things like demographics and vitals. There's also a lot of billing data. So all of this works to support actually getting paid for the care that's provided. So there's a lot of information on like diagnosis codes that are used for billing, not necessarily for clinical care. And as you know, something else to consider is that this is a legal record. So it's not just about your health, it's also maintaining legal custody over what care was provided. And so that comes with some unique caveats on how to work with EHR data. So how do we actually take what is a per patient view that I need to click through and read all these notes and actually make it usable for research? Well, that happens by going to an EHR-based repository, which here at CU is Health Data Compass. That's going to be your core data resource to work with EHR data here at CU. And then you can work with those data and leverage them for clinical trial recruitment. Let's say, you're running a clinical trial and you're not meeting enrollment numbers because it's been really hard in the pandemic. Working with EHR data can help give you a list of potentially eligible patients to approach and do so in a higher throughput manner than just relying on who's coming to your clinic each day. We can also leverage this data to build healthcare analytics or the learning health system. So let's actually use these data to learn about the optimal clinical care that we can provide patients and potentially also learn from the new innovations that we've implemented in clinical practice, how best to inform our care. Of course, there's plenty more applications, especially with the pandemic. There was a lot of work for using EHR data for population health and epidemiology to be able to track in real time how the different pandemic phases were hitting. So lots that we can do there to try and better understand what's happening at a population level based on these data. And finally, we can think about how we can link these data to biobanks to enable genomic research. So typically, genetic studies, not only do you have to measure all the genomics, you also have to measure all the phenotypes for these patients. And so you're really limited by your time and resources. With an EHR, we've got a lot of clinical information. We've got a lot of phenotypic data. And thanks to resources like the CCPM Biobank, we can actually then link that with genomic data to actually speed up that translational research and genomics research. So that all said, exciting frontier ahead, right? We can do so much with EHRs, but I need to warn you, if you're coming from a traditional research paradigm, let's say clinical trials or working with cohort studies, where you've got really sort of clean control over your data, you have to make a paradigm shift to work with EHR data. These data are not sparkling clean. They are not even rectangular a lot of the time. So, the thing to keep in mind is these are data that were meant for clinical care. And in many respects, it's actually just to support billing or legal operations, not even about the clinical care of the patient. And so you need to think about the fact that there's a lot of process that you have to put in on the analysis side to really take these data that were never meant for research and make them useful. That said, there are trends in these data. Just because it's messy and it's complicated and it's a lot of work doesn't mean that the data are all worthless or it's not going to show you anything that's meaningful or real. So just be aware, you're not going to have an analyzable data set right out of the gate. There's a lot of work to get there, but these data are valuable. They do serve a purpose and can be very useful for research. All right, so let's talk about that landscape. I like to think about EHR research on two parallel axes. The first is looking at the scope of the research. Are you trying to do broad-based, large-scale research, or do you have a really focused research question in mind? And then think about the analytic complexity. You may not be able to assess this right now, but we'll talk about it in a moment. So is your analysis something that's going to be very simple, or is it going to be a more complex type of statistical analysis? On the broad and simple side, we have things like phenome-wide association studies. So this is, as you can imagine, looking across the entire phenome of patients. So looking at all the diseases a patient might have, but doing so in fairly simple analysis way. So using essentially logistic regression. But it gives you this sort of really broad view of what's happening with the health of patients. Whereas on the other hand, there's a methodologic tool called computational phenotyping, which is a very focused scope. You're identifying a single cohort. I want to find all patients with reduced ejection fraction heart failure. And the methods to get there are far more complex for how do you actually define that outcome. And I know we always have a lot of folks that are really interested in how can I apply AI and machine learning and predictive analytics? And the reality is those tools can be applied really anywhere along this spectrum. So there are some analyses that are pretty straightforward in terms of the informatics design, so they have simple methods, but they may be more broad in application. So just be aware that that sort of falls in between. There's no true rule for where machine learning or those predictive analytics can lie across the spectrum. So another thing to keep in mind, we're going to go through the different types of data that are present in EHRs, but a good framework to be aware of is this idea of how are the data, what's the form that those data take? So you probably all are familiar and have experience working with things like structured data. So this is something that you might find in an Excel spreadsheet or a database. You've got rows, you've got columns, and there's single values in each of these cells. So in this case, things like diagnoses, labs, they are all structured data. You may not have had the opportunity to work with unstructured data, so things like clinical notes, where basically it's an empty text box and you can type whatever you want, and how to make that usable. Semi-structured is imagine the unstructured notes, but now make a template or an outline where you've got a sort of standardized form, but then within those sections, it can take any text. And so as we think about these types of data, you're going to see all of them in electronic health records. Those data that are structured are typically stored in databases, whereas those data that are semi-structured or unstructured are going to require more complex tools like natural language processing to actually put them into a structured form that are usable for research. So as we go through the different data types, I'm going to talk to you about whether they're structured or unstructured and how we can use them. So the most common data type that we see used in EHR-based research are ICD codes or diagnosis codes. ICD stands for International Classification of Disease. In the US, we actually use what's called ICD-10-CM or clinical modification. So typically ICD is actually used for death statistics reporting internationally and some healthcare uses as well. And so, however, in the United States, we use these primarily for billing. And so Center for Medicare and Medicaid Services have created this clinical modification that is not quite the same as ICD-10 International. We did used to use ICD-9-CM. We made that transition in October 2015. So as you're working with clinical data from Compass, depending upon sort of what time span you're looking at, you may need to consider doing definitions with both ICD-9 and ICD-10 codes. Importantly, ICD-9 had about 14,000 codes. ICD-10 was a dramatic expansion. It went to more than 70,000 codes. And you may have heard some funny stories about this. So they got really, really specific. One of my favorites is W59.22, struck by turtle, subsequent encounter. They, in fact, make coffee table books with illustrations of the zaniest ICD-10 codes. So while it is more specific, not every clinical condition has an ICD-10 code. And so recognize that there are some limitations. So let's dive into those. ICD codes can have false positives. So one thing to know about medicine is that diagnoses evolve over time. So as a patient presents to a clinician, they're going to essentially make a hypothesis about what the patient might have. And they need to test that hypothesis by running laboratory tests and other tests to actually see what's going on. And so they're going to bill for the suspected diagnosis at the outset. And as they get more information, that diagnosis may shift as you reveal more information. And so you'll see this sort of code drift over time. Of course, there's always the opportunity for errors. So sometimes the wrong code gets entered by accident, or in a lot of cases, the workflow is such that it's easier to find one code than another, or historically with outpatient providers, they're entering this in while they're going through, and they didn't necessarily in the old days have the same sort of search functionalities. So they might have just had a handful of ICD codes that they have memorized that most patients have and use those instead. So there can be those errors. And it's important to recognize that due to how healthcare is delivered in the United States, there's also times where you're just required to put the wrong diagnosis because that's the only diagnosis insurance will pay for. So there's a number of treatments for psoriatic arthritis that insurance simply won't reimburse, but they will if you have rheumatoid arthritis. And so you'll see patients get diagnosis codes that really have more to do with this insurance billing than anything else. Questions in the chat. I'm going to defer some of them to later. Some of them I will answer now. So do ICD-9 codes still exist in the EHR prior to ICD-10 coding implementation? Yes, they do. So those codes may still be represented as ICD-9 codes. So you do need to take them both into account. And Jed, your question is great. I'm going to save that for the end. All right, so like I said, physicians may bill for different conditions. But in addition to these false positives, we also have false negatives with our diagnosis codes. So this is less true today, and it may depend on your data sources and sort of what time period you're looking at. But outpatient billing historically was limited to 4 diagnoses per visit from a billing perspective. And so you may not have all of the diagnoses that a patient has. Even if there isn't that billing, obviously providers are limited in time. So you may not get a full disclosure of all of the conditions that a patient has, just the ones that were being treated in that condition. Typically, your outpatient billing is done by physicians themselves. And so you may see some coding biases because it's too hard to look up all of the 70,000 codes and figure out the optimal one to use for each patient. Inpatient billing actually comes with some other biases, which is typically that's done by professional coders. So they're going to omit codes that don't pay well, that don't have high reimbursement. And they're also limited because they can only code for problems that are explicitly mentioned in the documentation. So oftentimes the clinicians in the room may have received friendly messages from their billing department saying, hey, could you make this a little bit more clear on your notes so that we can appropriately bill in this manner and get the appropriate reimbursement that we need to care for these patients. So just be aware, diagnosis codes, easy, helpful, but not always perfectly accurate. And the accuracy is really going to depend on the code in question. So for example, tobacco use codes, really specific. If you have a tobacco use ICD code, you almost certainly are a current or former smoker. But some of the work that I did showed that only about 30% of smokers actually get one of these codes. So very specific, not very sensitive. So just something to keep in mind. Another common data type that we use is laboratory data. These are structured data. There's a lot of positives working with lab data. It's closer to the biology. So if you're one of the more biomedical folks, it may be very useful. They often can be continuous variables, which means from a statistical perspective, we're going to have more power. There's no controls that we have to deal with. It also opens up some interesting analytic designs where you can look at the extremes and actually use that to drive innovation and really understand sort of what happens at these physiologically rare but possible edges. But there are some challenges. So you tend to have multiple panels that have the same labs. So just from an analytic complexity, getting down to the exact laboratory test that you're interested in can be complicated. It usually takes a lot of effort. It also is the case that people tend to have multiple lab values for any given test. And so the question is, which one of those should you use? The biggest, the smallest, the median, the average? Should you filter some out based on context? Lots of analytic decisions that you have to make. Importantly, the method and reference range changes over time. So we've had the EHR here at UC Health since about 2011. So that's over a decade worth of laboratory test assays that have been in place. And so they're going to shift whether you use one methodology or another. Those are going to come with different reference ranges. And so as you're doing these sort of long-term assessments of patients, you're going to need to think about how to handle those shifts. Units are always such a pain. Oh my goodness. So making sure that they even have the units, checking the units, because as those reference ranges change, as the methods change, they may actually have different units that they're reporting. And so again, harmonizing all those values across your study are going to be important. And there's finally a lot of junk information. So a lot of these results may not actually even be the format that you're expecting because it's a little bit of the Wild West. So just be aware, like, it's good, it's useful, but it definitely takes some effort to get through. So with medications, another common data type that you'll see in use. So typically nowadays, these are all electronically prescribed, which is great. So you're going to get things like the name of the drug, the size, the frequency. Those are typically structured, though not always. Dosing instructions like take 2 pills twice a day usually are semi or unstructured. So you're going to have to do some processing to make sure that you're understanding the full total dose that a patient is getting based on those instructions. You may also find drug medication information in the problem list and clinical notes. Both of those are going to come in unstructured forms, and so you're going to have to do some of that natural language processing to make use of those data. Some of the challenges is just recognizing that there's a lot of different names for drugs. So atorvastatin and Lipitor are the same thing. One's the generic name and Lipitor is the brand name. There's also drug combinations. So Liptruzit is atorvastatin and ezetimibe. And so you want to think through, okay, I'm looking for Lipitor. I need to consider its generic name. I also need to consider any of these combo therapies, and also recognizing that there's entire drug classes. So maybe I don't just want Lipitor, I want all HMG-CoA reductase inhibitors, including atorvastatin, simvastatin, rosuvastatin, et cetera. Or maybe what I'm really interested in is getting anything that's a cholesterol lowering drug. So I'm fine with the statins or ezetimibe, or fenofibrates. And the other thing to keep in mind that's always important to remember the context that you're working with. Just because a medication was ordered by a provider does not mean that the patient actually was able to fill that medication. They may have gone to Walgreens and been told, yeah, that'll be $300, and they're at the end of the month and they don't have that money to fill that prescription. So they may never even pick it up from the pharmacy. And even if they do get it filled, medication adherence is sometimes low. There may be reasons why they're not taking the drug, even if they have access. So just recognize that this is telling you something about the clinical thought process of the drugs that they are recommending. But if you're actually trying to say what effect a drug has, it may not be perfect because you don't really have an ability to tell whether the patient is actually taking the drug unless you're in an inpatient context. And so, in those cases, you may actually have nurse X gave it to the patient on Y date, and so we know that the patient got that medication in that time period. Another thing to keep in mind is that identifying discontinuation is difficult. So just because you don't have a prescription anymore doesn't mean the patient isn't taking the drug. And similarly, just because the patient still has an active prescription doesn't necessarily mean that they're taking it. Alyssa, I'll go ahead and handle that question at the end. All right, so the last type of data that we're going to talk about is unstructured data in clinical text. And so usually your goal, if you need data, information that's in unstructured data, is your goal is to take all that unstructured text and turn it into structured data. There's a lot of NLP tools out there that you can use. One of the most common is something called concept indexing. And the idea is to essentially take something like mad cow disease, map it onto a standardized terminology. In this case, this is a concept unique identifier, or CUI. And that CUI is going to be the same as bovine spongiform encephalopathy. So the idea is, hey, these are two different ways or representations of the same clinical concept. And so by mapping them onto the structured terminology, we can understand what it's actually telling us. Best NLP tools need to have negation or certainty tagging so that no evidence of diabetes doesn't result in you concluding that this patient is diabetic simply because the term diabetes was mentioned. Similarly, temporal contexts are important, understanding sort of when things happen, family history, all of those are really important things for you to consider when you're deploying an NLP tool or building your own. So that's just a brief overview of all the types of data that you can expect in an EHR that you might want to use for research. So thinking back then to this landscape, let's really go ahead and deep dive into two of these methods that I find are commonly used with EHR data. So let's dive into computational phenotyping first. So for those of you that are more genetics focused, you probably have heard of phenotypes and you're thinking about phenotypes as translating the observed characteristics or manifestations of human conditions and diseases. Or there's phenotyping in mice. Anything that's sort of this observable characteristic, that's phenotyping writ large. Computational phenotyping in my world is how can I use all this clinical data from the electronic medical record to identify subjects that are meeting some definition. So maybe I want to build a computational phenotype for diabetes. How do I identify all the patients with diabetes? Or how do I identify all the patients who have diabetes and an A1C well beyond sort of a good threshold? So they have uncontrolled diabetes, something like that. So in general, as we think about computational phenotyping, a common paradigm is you want to identify cases. In some instances, you may also want to identify controls for your study design. The approach then is build an algorithm that will detect some number of cases. So these are definite cases. We know that this algorithm has a high specificity, identifies these patients with great accuracy. Typically, we write another algorithm that will detect our controls with high specificity. Then there's usually some gray zone. So there's people that meet some of the definition for a case, but maybe not all of them. And these might be people depending upon the overall study size that you need for your analysis. You might do manual review to try and add those back in. And then there's usually a whole chunk of people in the middle that you exclude. So either they have things that kind of look like the condition, or let's say you're doing a study of autoimmune conditions. You don't want to include someone with a different autoimmune condition as a control if you're doing a genomic analysis, because they probably have a shared biological etiology, so they would not be a good control. So they get excluded from the analysis. Or let's say it's just a patient that came in, they broke their leg in the emergency room, and that's literally all you know about them. You may not have enough information to feel confident that they're either a case or a control, and so they get excluded. As you think about phenotyping algorithms, typically you go through a staged process. So the first step is defining your population. And by that, I mean, who are you drawing from? So here at Colorado, you might be drawing patients from UC Health. I want everyone that's being treated in UC Health. Or maybe you're a pediatrician, and so you want to look just at children's hospital patients. Or maybe you want to do omics analyses. So you're only going to look at CCPM Biobank participants. So sort of think of this as, what's the bucket that I'm drawing from? From there, you define your inclusion criteria, something that your entire population must have. And exclusion criteria, things that your population can't have. So common exclusion criteria would be, or inclusion would be, I only want adults. I want to exclude children, things like that. And from there, that gives you sort of a narrower subset of that broader population that you're working with. And from there, then you actually apply your final case control definitions. So let's say an example. Steroid-induced osteonecrosis was a phenotype developed at Vanderbilt. So in that particular instance, they were using their biobank. So their population is Vanderbilt BioView. So that's analogous to the CCPM biobank. And in that population, they required everyone to have some corticosteroid exposure. And it had to be something where it got into their full body system, so IV, IM, or oral, not an inhaled corticosteroid like if you have asthma. And you had to have a sufficiently long exposure to that medication to actually be able to see some of these systemic effects. They wanted to exclude things that can cause osteonecrosis all on their own, so that you wouldn't have sort of something that might be confused for steroid-induced osteonecrosis that's just due to some other clinical condition. And so this then becomes their overall study population that they're working with. And then they defined their cases with either an ICD-9 code. This is an older phenotype definition. So they only had that ICD-9 code for steroid-induced osteonecrosis, or if they had keywords indicative of that phenotype. And so for controls, they said, you can't be any of those things that are cases, and you can't have this record of another drug that would make them an unsuitable control. And so I want to bring your attention to a couple of things. Notice that this algorithm used medications. They had to have a corticosteroid exposure. It also used a billing code, or that diagnosis code. They also had some keywords working with clinical notes, and not shown here, but oftentimes will rely on laboratory tests. And what phenotype algorithms really rely on is using this combination of multiple data types. And it's only at the intersection of those data types that actually reveal the true cases. So people that you can be confident really have the condition that you're interested in. And so, of course, the question is, how well do you know that it worked? And generally speaking, the best approach for a phenotyping algorithm is to do a validation. So what you're going to do, you're going to take that data, you're going to randomly review a subset of records and develop your gold standard population, and just use simple statistics like your 2x2 table to assess your sensitivity, specificity, positive predictive value, et cetera. And that will give you a sense of how accurately am I identifying my patient population of interest. And then as you think about your controls, I will just warn you, defining controls is really, really hard because there's lots of different ways you can think about controls. Are they clinical controls? So you want someone who definitely doesn't have the condition, but they were assessed for it. That would not make a good biological control because they had something about their clinical presentation that made them look like they had the condition that you're interested in, but then the testing revealed that they didn't. But from a biological perspective, they have a lot of those shared symptomologies, and so they might not be the best to consider a biological control. So let's step back to that EHR-based research landscape. That gave you that deep, narrow view. The next view is looking at phenome-wide association studies. And so this is predicated on the view of, if you've ever heard of a genome-wide association study, where you have a single phenotype and you've got millions of variants in your genome. A PheWAS says, wait a minute, I've got all these phenotypes, so I can take some genetic marker of interest, and instead of assessing all the genotypes, I can look at all these different phenotypes that are represented. And so you can make a Manhattan plot just like you would in a GWAS study, but now each of those dots are phenotypes rather than genetic markers. And so this work was created by Josh Denny at Vanderbilt and has been expanded by Wei-Chi Wei at Vanderbilt, and is really a useful tool. And answer Paul's question of how can you identify patients who are part of CCPM Biobank, it is something that you can access, you can request from Compass. So you can either filter to biobank participants, or if you want to access biobank data, there is an approval process where you need to go through the Accessing Biobank Committee. But if you say you fill in a Compass intake form that says that you want to do biobank studies, they will link you out to ABC and talk to you through that process. So PheWAS typically is used for these omics studies. It can be used when you, let's say, have, let's say you're a basic scientist and you have some cool new mutation or genetic variation that you're interested in. You think it's important, all your studies found that it was really had some unique biological properties, but you're not really sure how those manifest in humans. PheWAS will be a great way to sort of do this hypothesis generation scan to say what sort of phenotypic consequence might be happening based on this genetic marker. It also is obviously a very key tool for detecting pleiotropy, where you might see a particular marker having different effects across different organ systems. So how does it actually work in practice? Remember that whole simple analysis? Compared to a computational phenotyping algorithm where you have to go through all these steps and use all these different kinds of data, PheWAS relies only on diagnosis codes. So it takes those ICD codes in the EHR, and there's a lot of codes that mean roughly the same thing. So 556 ulcerative colitis versus left-sided ulcerative colitis versus unspecified ulcerative colitis. Why do I have so many of them? Can't I just represent that as a single Phe code, ulcerative colitis? The thing that PheWAS does take into account is recognizing that, some of these codes aren't as good as others. And so you can do some numeric thresholds. You need to have 2 or more codes or 3 or more codes. And so you can imagine any individual patient, this patient might have 4 instances of 556.0, 1 of 556.5, and 7 of that unspecified code. And so when I roll all those up into the single Phe code, I've got 12 instances of that particular Phe code. And you can do that with the next trait. So anemia in the mother, anemia associated with pregnancy, so on and so forth. So the way that this functionally works in practice is for any particular Phe code, you say, do you have any of the ICD codes that map onto this trait of interest? If they do, then you ask, okay, yeah, but I want to be really sure that they have this trait, or at least mostly sure. So do they have 2 or more? If they do, then you call them a case. If they don't, so they only had 1 of those Phe codes of interest, then you exclude them. You say, they've got some indication that they might have this trait, so I don't want to call them a control, but they don't have enough evidence for me to be sure about it. So I'm going to simply exclude them from that Phe code. If, let's say, they didn't have the ICD code at all, Then we ask ourselves, do they have any exclusions? So we talked about this a little bit or alluded to it. Let's say I'm looking at a fee code for rheumatoid arthritis. Because PFAS started in omics analyses, we're thinking from a biological control context. So if I'm doing an analysis looking at rheumatoid arthritis, I would not want to include someone with lupus as a control because they have a shared autoimmune background. And so if I have one of those exclusion codes, I'm going to exclude them from the analysis for that fee code as well. If they have neither the ICD code and none of the exclusions, then I call them for a control. And then I do this for each of about 1,400 different phenotypes. There's actually an R package that does this for you. We also do have all of these implemented for Health Data Compass, or at least for the biobank. We're working on generating it for all of Health Data Compass. So if you're trying to do something for non-biobank participants, highly recommend using the R package. It has translation tables for all the different terminologies that you might want to use. It also has analysis scripts, can do multiple testing, correction, and a lot of really nice actual analysis for you. If, however, you want to do omics-based analysis with PFAS, we actually have all of those pre-generated thanks to the translational informatics services. So my group generated fee codes. They ran all of the analyses for all of the genetic variants that we have in the first and soon to be second freeze of the CCPM biobank. So if you're just interested for, let's say, a grant proposal or something like that, you can actually go look at that information. We do have a new browser that the Translational Informatics Service Corps has brought up. And so that's, again, something that you can access, I believe, through the Accessing Biobank Committee. So that's a great resource. But importantly, just because you get a significant finding does not mean that that necessarily is a true finding. So one of the things that you'll see is that PFAS has a lot of code correlation. So just because there's a significant hit doesn't mean that that's really the driver of the phenotypic association. So a great example of this, when I was in grad school, did an association, and I found one of my SNPs associated with the fee code, acquired loss of hair. Turns out this is a SNP associated with breast cancer risk. And so it is not associated with acquired loss of hair. It's associated with breast cancer, and women with breast cancer go on chemo, and chemo results in them losing their hair. And so importantly, just because you get a hit doesn't mean that it's real. You have to do a lot of work to take those association findings. So look at your genotype and phenotype frequencies. Ask yourself, is it plausible? Is it meaningful? Does it make sense biologically? Actually look at the individual patients with the code and their records to understand sort of what the driver and the follower codes might be. And always remember that fee codes are phenotype-like. They tell you something, but they are not nearly as robust as one of those computable phenotype definitions. All right, this is a hypothesis generation tool. It's really useful to give you a direction of where might signal be, what should I follow up, but you do need to go in and do those follow-up analyses. And so move from PFAS to a computable phenotype to validate or do these sort of other confirmatory steps. So with that brief tour through the EHR-based research landscape, I want to give you some resources that you can use here on campus. So we already talked about the fact that Compass is our EHR repository here on campus. They have a number of self-service tools, as well as the CCTSI has a number of tools that can be useful if you're trying to do clinical trial recruitment. So we have Trinetics as a tool that you can use to identify patients who might be eligible for your trial. And we're having a new tool that's being brought up soon called Leaf that can also be used for that purpose. Obviously, REDCap is a wonderful tool. I use it all the time for doing actually that chart review validation step, but it's also a great tool for clinical trial recruitment. Compass can actually send you trial recruitment potential possibilities for like, here's all the people that meet your condition, and actually return them into a REDCap database for you to approach. So that's a great resource. If you want to do population health epidemiology or really any of these, CETA is a wonderful resource. We've got a lot of biostatisticians and data scientists who have been working with COMPASS data, who know what they're doing, who can really help you design an optimal study and get down to what specific data elements do you actually need to answer your questions. In terms of learning health system and analytics, we have Data Science to Value program, the D2V program, the new Center for Health AI and Department of Biomedical Informatics. Lots of great folks who can do really advanced modeling with EHR data. And like I've been alluding to throughout the talk, the Colorado Center for Personalized Medicine Biobank is a huge resource for campus to be able to do genomic research. If you want to learn more skills, more than just this quick breeze, I do have a number of resources available. So if you're interested in sort of the study design process, let's say you're a PI, you know you're not going to be the one hands on a keyboard, but you need to understand how to write a grant or how to design your analytic plan. I do have a CETA short course. The schedule is still being determined, but it will be sort of early spring semester. And it's a 6-week short course, no credit, but you do get a certificate of completion. You don't have to program it. Show up, talk, think about your study design, and do a little bit of a deeper dive. I also have a course that teaches analytic techniques. It's by 6310, Practical Clinical Research Informatics, online, asynchronous only, and it's 3 credit hours, but this one does have some programming prereqs. Unfortunately, I'm not offering it this coming spring. I will be offering it again in spring of 2024. So that's a long time away, I know. So if you want to get that information now, it's also available as part of my Coursera Clinical Data Science specialization, which everyone can access for free if you're a CU faculty, staff, or student. Just go into the mycu.edu or just look up CU1 Coursera, and you'll find the instructions for your group. And that has all the same information as Bio 6310, but you're doing it completely on your own versus in course. I'm available and can answer questions and things like that. So these slides will be posted. So I do have some links for you that you can look up additional information, and happy to take questions. I'm going to go quickly through and talk about some of the questions that came through that I chose to skip through. And go from there, but feel free to ask any additional questions in the chat now. But thank you all so much for your attention.
Thank you so much, Laura.
Absolutely. All right, so back at the beginning, Jed had a great question. Are drug-drug interactions annotated in the EHR? There are a couple of answers to that question. So first, are drug-drug interactions listed so that someone can search for them? As a researcher, no. As a clinician, there are sometimes alerts and things that will fire saying, hey, this drug has an interaction. So there may be some clinical decision support to help providers along the way. If you're asking, can I just go through and find patients who have drug-drug interactions because I can just like search a structured data field? No. Typically, you may be able to find some adverse event reactions annotated in the allergy section, but usually drug-drug interactions or any sort of adverse event detection is going to have to happen through natural language processing. All right, and then Alyssa asked, can you discuss a little bit about the data quality of outpatient medication dispensing fills in Health Data Compass? So there are some, so patients that are getting their meds getting their meds filled through one of the pharmacies at UC Health, we should have fill data there, but I would say that the vast majority of patients get their medications filled elsewhere, either through their insurance, mail order pharmacy, Target, CVS, Walgreens, et cetera. So, you know, outpatient medication dispensing is going to be still a pretty limited data source. And then Alyssa asked another question, can we still do computational phenotyping if manual chart review is not possible? So you're doing de-identified, I'm using de-identified data. you can still do computational phenotyping. Like, you can still apply the methods. You can take an algorithm from the literature and apply it in practice, usually. You simply won't know if it worked or not. So depending upon your study design and how robust it is to patient misclassification, that can be fine, or it can be not fine. And you're just going to have to assume that it's okay, unfortunately. So you can do it, you just aren't really sure how well it worked. That's a great question.
Great. Thank you so much, Laura.
Absolutely. Any other questions that the room has? All right. Well, if you do have questions along the way, would love to see you in one of my courses. Always happy to chat, and make use of the various resources that we have on campus with Healthy to Compass, with CETA. Lots and lots of really knowledgeable folks on campus that can help you with these kinds of studies. Lots of opportunities to learn. So definitely take advantage and continue coming to this wonderful series that goes through lots of different methodologies and gives you a nice overview of approaches that you can take. So with that, I hope you all have a great